Welcome to Tech Talks. I'm Holly Scott. Today I have the distinct pleasure of speaking with Frank Bonnet. He is the CEO and co-founder of Bionomus. Frank received his master's and PhD from the prestigious EPFL University in Switzerland, where he studied robotics, mechatronics, and automation before starting Bionomus in 2019. Thanks for joining us today, Frank.
Thank you very much for having me. It's a pleasure to be here.
Likewise, it's definitely a pleasure. It's been great getting to know you through the course of our work together, and really interesting and excited to chat further about the Bionomus technology. So great to have you. So let's start there. You know, this is a really interesting problem that you looked to solve here with Bionomus. Can you tell us a little bit about the origination and the thought behind starting this business?
Yes, so it actually originated during my PhD at the EPFL that you mentioned. So I was during my PhD, I was developing a robotic fish to study the behavior of small species of fish, such as the zebrafish. I will explain a bit later about why does this fish really important for research. But so the goal was really to study how the fish behave in a group, how do they take decisions, and to have a robot mimic the behavior of the fish, so that it's kind of a spy inside the fish society. So that was the goal of my PhD. So I developed this robot, and I was setting up. My goal was to installing it at the lab of biologists, so that they would perform the studies. But every time, so the biologists were based in Paris, in France, and every time I was going there, when I arrived, it was in the morning, and what I saw is that instead of seeing them using the robots for the experiments, they were sorting the eggs of this fish. And I was like, why are you doing that? You know, it seems to take a lot of time. But they say, no, it's for this fish is actually used as a model for research, and actually it's mostly the embryos that are used. And so to breed them or to make some experiments, we need to sort these eggs, screen them, sort them, pipette them by hand. And so it started as a joke. I told them, okay, I will also make a robot to do that, so that you can spend more time with the fish robot to produce data for my thesis. So it started a bit like this. And so little by little, we started in the lab to say, okay, we have this idea, so let's try to make a robot that will automatically screen and sort these small fish eggs. And because I was also in this place, like where doing my PhD, I was going in conferences that we specialize in the studies of this fish. I saw that it was a need that was actually not only from this lab, but from like all the people working with this model. And so this is why we decided to go further, and we got some financing, and this allows us to further develop the technology and then found the company.
That is fascinating. I didn't know much about it until we started working together and really looking more into it. But the zebrafish, and I know that your technology has applications in other entities and biologic entities, but zebrafish in particular is of high interest, specifically in research. Isn't there some interesting properties of the zebrafish that allow for pretty strong study? Is that correct?
Yes. So first of all, it's a very interesting model because it comes as a replacement of some other traditional animals, such as the mice, for instance. We all know that mice, they are used to test new drugs before they are kind of implemented in human bodies or become real treatments. But actually, what they discovered is that the fish, so they still are sharing 70% of genetic material with humans, but there are 84% of diseases that human diseases that have a zebrafish counterpart. So actually, the zebrafish in terms of genetic material can be used also as a model for studying a lot of disease that we have, or for instance, cancer, heart disease, or also like some other more like genetic research on genetics, for instance. And this model has an interesting property is that these fish, they produce these eggs, and so what happens is that you have every 2 or 3 days you have 100 of eggs that are produced by one female, so you have a lot of samples compared to mice. And all these eggs, they grow outside of the adults because the embryos grows outside, and so you can see the embryos developing. They are also transparent, so you can see all the organ developments. And so this is why actually most of the research is done on the embryos at very early stage, which allows to be very like it kind of goes towards this kind of automation of research, because you cannot do on mice with the zebrafish model. You can in several days you can already have a lot of data of drug screening, or trying to maybe see the effect of a genetic mutation, for instance. And so this allows you to have kind of high throughput research with this model, which you cannot have, for instance, with the mice.
Wow. So there's a strong compatibility from a scientific and genetic standpoint. So
yeah, exactly.
Did you say 80+% genetic similarities in terms of testing properties?
In terms of diseases, for instance, that you can test in both kind of human and zebrafish are the same. So of course there are some that we don't share, but there are a lot of them that you can actually use the zebrafish to study. They are also used for diagnostics of disease such as cancer more and more. And so this is why, and this is where the research is going. It becomes a model that can be used for several applications. But because now the goal is also is to really apply this, for instance, for diagnostic of cancer, we need like the researcher or then the companies that are using this model, they need tools to automate some of the process, such as the screening, the sorting, the pipetting, and all of that. And that's why it's now a demand. Not the zebrafish is a very established model, but now the kind of this. The use of this model requires also more tools to automate some of the processes that are done with this model to have costs that are affordable so that it can become even more popular.
Mm-hmm. That was impressive too. When I was looking at the comparison, the current standard of care, it looks like it's not only highly manual and time time consuming. They're using microscopes now. Is that without it? So do they have to look at every single zebrafish and determine if they're...
That's the thing.
Oh my goodness, wow.
That's the thing. And sometimes they have to just sit down for 2 or 3 hours in the dark room, because they look also in for fluorescent signals. Like for instance, they want to track the growth of an organs, for instance, and so they need to look for that and all that. Yeah, you need to, if you for certain certain research, you need really to individually screen your eggs. Then you will do something with these embryos, label them, plate them, or anything, and so all that of course takes a lot of time, and it's not really really cool to sit for 3 hours in the dark room with a microscope. And sometimes it's even the professors in the lab, or like even people with high degree that are doing that in the lab for their research, and they have to spend a lot of time to do this job. And if you look more into industry also, because some industries are also adopted this model, then it's more, of course, it's more like will be technicians that will do this job, but then it's a job that takes really the full time of a person for every day, like an everyday job. And so the goal is also to make this as efficient and also as efficient as possible, and also reduce the cost of these companies, so they can really provide have a strong services that they can provide.
Well, that's a completely appropriate way of looking at the fit with robotics in the world, right? The world of work. And there's this big theory, the robots are going to come take over the world. They're taking all of our jobs and such. But the reality is, if we can automate the segment of work that it doesn't require, it's time consuming, and it doesn't require a higher level of thinking, it allows for the individuals who are right now using hours upon hours upon days to go do higher level research. They can now get to the meat of what they're trying to do sooner. And that's the beauty of this type of innovation.
And I think what we bring also is the fact that for of course there are some applications where the people would have to screen all these entities one by one, but sometimes they don't even really spend the time to doing that, because it takes time. And so with our tool, we can also kind of generate more data actually that the user will do. So all this data, of course, can be also useful to even enhance some of the research they are doing, or like discover something that they would not even think of if they would have done this by hand, or because they would have done it by hand, they would say, oh, I will not take so much time to do this sorting like super accurately, or like screen all my eggs. But by doing that, you allow that this is something we are still discovering because we are deploying our product, but we see that we can also produce data that could be also very valuable for the researchers.
Yeah, and that's fascinating. Let's talk more about that data component, because I know that there is an interesting, it's a screening, sorting, and dispensing technology. So from a technological perspective, can you walk us through that, how it works and what those components look like?
Yeah, so in terms of the device, so you have to imagine like big printers or something like well, something that can still fit on a desk or a lab bench. And so the components inside the device, of course, you have some mechanical components. You have fluidics because everything is with working with water, and you have a bit of electronics and an interface like so this, just to picture you like what what the instrument looks like. And then basically the process is the user will come with 1,000 eggs or 1,000 seeds, or because we are not capable of using it for different types of small entities. So you will place them inside the device. So there is a kind of small hole, so you place them inside this, and then the device, what it will do, it will through that the core technology will allow really to individually grab these entities. So that's the first part, like from 1,000 entities, we'll be able to take them one by one, select them. And then that's the part of placing them in front of a camera. So how to have a kind of image where the entity is always placed at the right position. So, and then the first time you would use a device for a new application, we would of course train an algorithm. So based on these images, the user will have to label them and then train an algorithm so that the device will kind of learn this is a good seed, a bad seed, an egg that is fertilized or not fertilized, and then based on that the device will do like pipette them, plate them, just output them into Falcon tubes or any types of output like container that are used in research, and so that's basically how it works. And so the goal is that the device needs to operate, like do a certain series of tasks that are done by humans, and trying to be as efficient as possible in terms of efficiency, like precision, but also of course be faster than humans. For the moment we are precise, but we are in the range of humans. So for the moment it's kind of replace all the work of humans, but now our goal is really like over the years to improve the accuracy of the speed of the device so that it can even outperform humans.
That's great. There's some real opportunity there in terms of potential. I was impressed by the footprint too, because even as a first generation technology, in most clinical settings, real estate, the size is quite important, because there's lots going on in the lab, there's lots of other things going on in a clinic. It's really not that large, especially compared to you have the traditional setup involving microscope and other instruments that are around. So I appreciate that, because that's not often when you have such a complex inner workings with everything going on inside the instrument. It's hard to keep it to a certain size.
Yeah, so we tried. So really at Bionomous, we are really market driven. So really we try to build the instruments that our user wants. And one of the requirements of the user was this has to fit in a lab bench that is usually, as you said, kind of crowded with other things. So we know that sometimes some equipments, they need also an external computers, etc., and it starts to take a lot of space. So our goal was really to kind of have something where everything is embedded and doesn't take a lot of space. Of course, it was a nice constraint, I would say, for the engineers at Bionomous to kind of fit everything inside this. But we know that this is the types of equipment that our user wanted, and so we try to make this we said as portable as possible, even if the user, they don't transport it every day in the lab, but let's say as something that you can even in one day just take it in one place and put it in another table, or if you need space, and that doesn't take too much space on the lab bench, because you need also to have your tools around.
That's interesting. Yeah, that's a really interesting point. So with the throughput right now, and again, your first generation, I know obviously you're going to be improving on it. What does it allow for the standard researcher to do from an advantage perspective? So, for instance, today, Tom scientist sits down and he does X number of embryos in certain number of hours and gets X% accuracy versus what we're seeing right now with your Bionomus results. Can you share a little bit there?
So this is probably the one of the hardest questions we have now, because the numbers of applications we have are already so broad that it's very hard to to have an exact, like a very representative one, because we have some cases, for instance, where the device is actually for like maybe very basic sorting, the device would be the same time as a researcher, or even some others, because some of the researchers are super well trained, so even sometimes even slower. So that would be like, for instance, if you do basic bright field sorting, but then if you go to fluorescence, for instance, I was mentioning that before, then the device outperform the humans, like can go 2 or 3 times faster than the humans. So that's why it depends a lot on the applications. And then regarding accuracy, it's the same. So it also depends on the application, but also on the on the technicians, the quality, the efficiency of the technician you are mentioning. So but the device is true that we guarantee accuracy of 98%. This is like for the full process, including the sorting. And of course, maybe sometime a very well-trained technician that has 30 years of experience will maybe outperform that, but the basic researcher or technicians, usually they are not that high. So so this is why we can guarantee already a bigger precision. Then regarding the speed, for the moment, what we guarantee is that on average we are at the same time. So it's really something that the researcher, instead of spending now 2 hours for sorting, he will be able to do something else, or process data or whatever, while the device is doing that. So really relieve fully the time of the technician. And now also we're trying to also increase this efficiency. This is maybe more for the industry applications, where people will also want to, of course, improve the cost of of all their process, and also their throughput.
Excellent, excellent. And that's a fascinating point when you think about what you're moving towards. So if you're starting, this is only first gen, and you already can get the same or better results in the same or slightly better time. You're able to be more comprehensive with the data grab. So you're using this AI, and it's collecting information along the way, correct? So in terms of so like any AI device, it's getting smarter, and it's getting it's getting more and more powerful. So I could see that continuing to be a growing benefit.
Exactly. So we are really in the phase where we are learning with our customers, also with these new applications. So I think every month we have someone else contacting us saying, "Oh, I have this application. Does it work?" And sometimes we say, "Oh, not at all." But sometimes it's actually super interesting, and it requires only maybe a small software change or a very small hardware change to kind of have the device running for these applications. And so it's really exciting. At the same time, of course, it's, as a startup, you're trying to maybe focus on the core application, but we see a lot of potential. So it's also a nice way of exploring now some very high potential application that could be the future for us.
Yeah, I can imagine quite a number of applications which could be the future. So I know one of the biggest challenges I've spoken with startup founders on is sometimes focusing and staying clear on what you're going to go for first. Go here and get that penetration, and not spreading yourself too thin, right? So. But I have to come back to one area because I think it's really interesting. So you founded this based on a bit of a joke, right? You were working on your PhD, and you were creating this robotic fish, this spy fish you call, which I think sounds really cool in its own right. And you looked and said, "Boy, there's a gap in the market. You see this need. You see people doing this task every day, and how cumbersome it can be." So to me, that's quite entrepreneurial. Did you know you had an entrepreneurial bent? You're in a compelling education. Did you grow up in a household where there are entrepreneurs? I'm curious about where that interest in innovation came from.
So I can say that not at all. Like I have nobody entrepreneurial in my family. So I'm also very surprised. I was also very surprised. I can say that first, I think like the PhD helped a lot in because in the PhD you have your research that you need to kind of manage. Also, you have sometimes students that work for you. So it kind of emerged for me like this maybe entrepreneurial mindset about you have a project that you need to lead up to the end to get your PhD. You need to have maybe a small team to lead, and so that helped me. And also in my PhD, I had this robotic fish, so this pie fish. So it was already kind of a product. So that's really the idea. The goal of my PhD was really to build this product, make it as good as possible for the end user, which were kind of other researchers, and having a team around to be able to help me to achieve this. So this is why I think kind of created this entrepreneurial mindset for me. And then what I think was very valuable, and this is something, I don't know how it works in the US, but it's something that now in Switzerland they push a lot, is that if you are in this, if you have finished a PhD and you are in the lab and working, for instance, as a postdoc, if you have an idea, like the kind of technology transfer office will just say, okay, I give you, 50K, just have fun and trying to develop this. So it really worked like this. And so it's really helps, because of course, as a researcher, or like if you don't have this entrepreneurial mindset, you be maybe also are not willing to risk so much, because academia is so good, because you have your small small grant, etc. It's no stress, so it's all good. But then if you have someone that just give you a bit of something to say, okay, just have this and develop it, and you see that it's not, you don't need to take this 50K from your pocket and invest and all of that. And so now in Switzerland we also have a lot of support, like from government to create your company, and this helps really, like you learn from the PhD, and then you have just this support, like instead of just saying, okay, I give up, I go to industry, or I stay in academia, they say, oh, try to bring that to the market, and here's a little help for that. And that helped me to say, okay, I will do it and try, and we'll see.
Well, that's an incredible advantage. There are some programs in the U.S. that offer that. From where I sit, from a career development perspective, that's powerful, because so often, especially PhDs, especially people who are following a strong academic career, they often struggle moving into a development landscape where they're really going to put a product in the hands of a customer and drive it that way. And look, rightfully so, they're armed for perfection and for making sure that they've gotten everything out of their idea, where it sounds like what might have been an interesting challenge for you, as well as an opportunity, is the allotment of the investment allowed for you to think developmentally and say, okay, do we have a product that's good enough to start moving into the hands of customers so that they can start benefiting from it? And by that, you're going to learn more. So this is an iterative process overall. So I love the parallels that you gained from your PhD program. That's pretty interesting, that it allowed you to think like a startup entrepreneur in that program, and then coupled with the backing of the investment and allowing you to, really encouraging you, not just allowing you, but encouraging you to develop that idea. That's remarkable, because look, a company founded in 2019, which is already producing revenue, that has sales in the healthcare life sciences space, is pretty good. I mean, that's not something I see all the time. So tell us a little bit about that sales process for you, because now you're going from a PhD to the first salesman in Bionomus, right?
Yeah, exactly. So, and here again, I think, so I'm also like not really like originally more like an engineer and then a bit scientist through the PhD. So also, but I think also the sales process came during the PhD, because during the PhD you need also to kind of sell your research, because you need to publish, to have reviewer accepting your publication. You need to have maybe you have some of your colleagues that accept your products, because they so I kind of learned a bit, like on these aspects, and also, and I think what I I saw really like going in this conference, and like what inspired me really like creating Bonnet was going in this conference and see, in conferences you have these vendors that are the sponsors of the conference. They are here to sell the equipment, so it's really that the next steps after the research of the creating the equipment, and you need to go and sell. And so I got inspired by how they did what they were doing, and so it's quite inspired to say, okay, we can really create a company and sell products to these researchers. And of course it was a complicated transition, but I think as you become the CEO of the company, basically what I realized is that you have to sell every day, because you need to sell to your employees, you need to sell to all your stakeholders, your investors, etc. So it's again like something that you need to learn, and you need to learn how to do. And of course, sometimes it's a bit like I don't know, like a bit of schizophrenia, because of course you sell to everybody, but not exactly the same thing to everybody. So the sales process change a bit, but then you become as a CEO, I guess you become a salesperson all the time. So.
Well, that's great foundation, and it's allowed for you to have some early success, right? Your products, where are your products at right now globally?
So that's the good and the bad thing, I would say. So because we have really like a worldwide market, so so we have actually one device installed in Australia, one in China, like 4 in the US, and the rest in Europe, which is great because we see like the application. But of course now we see that it's also we took the risk of doing that because, for instance, and this is why we entered in contact, because we saw a strong traction for our product in the US. And so this is why we want to move forward with increasing our presence in the US in the near future. And so it was exciting at the beginning, but of course also a risk, because then we have devices that are in Oregon, which are probably the farthest place in the US we can go up, well, apart from Hawaii or Alaska, but still super far from Switzerland. So of course, when there is something that is broken there, like it's like, okay, I will need to do a small trip tomorrow to Oregon, so it's not super easy. But on the other hand, we decided we saw that there was attraction, and we decided to kind of take the risk and move forward with these people because we need I believe that when you build a product, you need really to have as soon as possible customer using the product, give you some data so that you can always improving it, so that it of course kind of helping your sales and development process.
Yeah, it's interesting. I was fortunate to partner with you on building this U.S. effort, and the population that I was chatting with was really excited about this technology. There's quite an interesting application. So for those of our listeners that are in the life sciences, definitely be on the lookout for Bionomus, because it's coming to us fast here, and that's great. It's a great opportunity for everybody. So we talked a little bit about where you've been and where you're at now. What are you thinking for over the next few years? What are some of the things you're looking forward to and expecting of the company? And also I'm interested in what you're excited about overall in terms of state of the industry and where the industry is going as a roboticist and as someone who's rooted now in science and application.
Yeah, so regarding Bionomus and the near future, so as you were explaining, so we started really like to commercialize the product end of last year. So it's really like we are still in, let's say, this sales growth phase where we in the first half of the year we had to build all the production, the stock, etc. So it's still like there was also quite growth of the company. We end of last year we were still 10, and now we are 25. So it's it's also trying to build the organization so that everything is running, operations are running, and we can also have all the team necessary to be with the customers and like distribute our products. So the goal is to keep like growing our sales in the next 2 years. So this is one of probably the core objective. But because we still have a big R&D team that was developing the project, we have 1 or 2 kind of technology that we are developing to have keep bringing new products in the future, where especially one also sorting the larvae of the zebrafish, so the stage after, and one more for industrial applications where it's more like high volume, high throughput. So typically for aquaculture, for instance, where they need to sort fish eggs, but really at a very large scales for fish production. So this is what we have now in the pipeline also for the future of the company. Because we see that not only the egg sorter or core product, but like the technology behind can be applied to several things. And so we want to keep exploiting that to the market. We see that have high potential. And yes, regarding robotics, so I totally like align with what you said before, like automation is more and more like I think the key, and well, we see it in all industries, I guess, and life science in particular. And yeah, I still believe that there will be more and more tools that automate some of the process that are done and that are complex to be done, and that also that are not efficient. And I think this keeps keep expanding and we'll keep doing this during the next years.
Yes, I think as humanity continues to get smarter and smarter, it only becomes opportunistic for us to create tools that can take off some of the manual work that is so time consuming. You know, there's just so much to be said for that. So it's exciting. It's a great time to be in the life sciences space for sure. And Frank, thank you so much for joining me today. This has been a great discussion. I'm excited about Bionomus and what we have in the future here. And to all of our audience, thank you for joining us on Tech Talks. Holly Scott, thanks again.